Good afternoon, everybody. I think it's afternoon for most people watching this presentation. And yeah, welcome to my presentation. My name is Werner De Gier. I am a PhD candidate at the Naturalis Biodiversity Center here in Leiden, the Netherlands, the Natural History Museum here. And I am here to tell you something about my last paper. So this is an ecomorphological review of Pcrabs. Yeah, so let's just start, I suppose. So, ecomorphology, a small introduction. Ecomorphology, as the name suggests, is a link between ecology and morphology. It's fairly simple, as you would see. The interesting part is that these ecology can come in all shapes and sizes. So, for example, habitat habituation, of course, behavioral ecology of these animals, but also dietary habits, all play a role in how an animal would interact with its environment. So the morphology part in this is more clear. It's often part in the exterior shape of the animal, but also the interior shape of the animal, so the anatomy, of course. The interesting part of ecomorphology and why I started to study it is the combination of very new and old methods. And it can go both ways. It can go in figuration methods, but also in analytical methods. So for example, in figuration methods, you see that you can figure an image an animal, so like for example the shrimp here on the side, in three different ways fairly easily, but highlighting different parts of its ecology or morphology. So for example, the illustration here is just for the exterior view. Videos and photos give us more interpretations on how the animal behaves in the wild. And for the same species, you can also, of course, put it in a CT scanner and color it different ways. So you can see also the internal structures of these animals and see if there are any traits in there. And something to remember during this presentation is that most of these ecomorphological studies tackle functional morphology, and oftentimes they're predictive research. So that means that if you study an animal very detailedly, how do we say that? If you know an animal very well and you see those same features in another animal, then you can say something about that animal as well. And that happens often in the higher animals. But we hope to do something like this in the future with my animals, which are Pcrabs. So a small introduction to Pcrabs. They are a true crab lineage. So they are located in the Decapoda Brachyura. And we call the family Pinnotheridae. And as the name suggests, they are very small. And that's not just because they are born small, well, they are born small as well, but it's mainly it has to do with their habitat. So they are considered endo or ectosymbionts. So they live inside or on top of another host, which I will go into more detail in a few following slides. But here you see already one of these Pcrabs inside of its host. Here's a fan shell, and this is an American species of crabs, Pinnotheridae. You see it already inside of the fan shell, but as you can see, only the female is located inside of here. The female is way bigger than the male, so there's a huge case of sexual dimorphism in this group. And the males are very small because they hop in and out of different hosts and are not often found inside of the host, while the females stay inside of the host in a sedentary lifestyle and they cannot leave their host ever again. There are more than 300 species recognized within this family, but most of them are subdivided into two major subfamilies, which are fairly easy to identify, as I will show you here. So this is a 3D model of one of the subfamilies, well, representatives of one of the subfamilies. Namely the Pinnotheridae, and this is my own focus right now in my research. And as you can see, there are a lot of characters which need some highlighting. So for example, you see the legs already there. Those are very thin, so the CT scanner didn't really see them that well. Very big, heavy claws. But if you look at the carapace, so the outer shell of the animal, you see already that it's really round. That's why they got the name Pcrabs, of course. But keep this in mind if I show you the other crab here. Because the other subfamily, the biggest subfamily, is the Pinnotheridae, and they're way broader. They are harder also as well, the harder sclerotized carapace. And you already see that they're fairly easy to identify. One of them is very round, the other one is very broad. So why? It has all the things has to do with host choice. So for example, here on the left, you see the species we just saw the CT scan of, but then inside of an ascidian. its natural host. They can also come in mussels, also in oysters. And here you see another species also living in a North Sea oyster. As you can see here, both of them live in soft hosts. Well, of course, the outer layer of a bivalve is very hard, but the inside is very soft. So they don't want to rub against that with a hard carapace. So they have developed a very round, soft carapace. While the other subfamily is found inside these tube-shaped habitats. So, for example, the holes of decapods, which bury in the sand. Also, a lot of kinds of worms. And for example, this worm makes its own little tube where a specialized crab lives inside of it, as you can see here on top. This can go to very extreme ways because some tubes are that narrow that some crabs even evolve themselves to be that broad that they can turn themselves over again in these tubes, as you can see here in this Peniculala, which is still undescribed. So we now know that these Pinnotheridae, so these globose subfamily, lives inside bivalves and ascidians, but there are way more hosts. For example, inside of these snails, inside of the body of these snails, so very much inside of these shells. In and also on different types of urchins, but also on the front doors or the back doors from different types of holoturians, so sea cucumbers. But are there more morphological characters which are linked to these different types of host interactions? Yes, but they're very subtle, which I will show you now. So this is, for example, an ectosymbiotic group of Pcrabs, so the Disodactylus genus. It lives on top of irregular urchins, but also on sand dollars, as you can see here already. And from a distance, you don't really recognize a lot of the characters which this crab has, because if you zoom into it, you see it's very hard, which is probably has to do with anti-predatorial characters. But if you look very closely at the legs of these top crab, you see that it has very small forked legs at the ends of its walking legs. So why does it have that? A few scientists think that it has to do with the way that they move around on top of these sea urchins, because that way they can hold themselves onto it with the currents and all the water flowing over them. A few very small characters. Another group with very subtle characters is, for example, the holoturian associated crabs. So, for example, this Brachyura is very hairy, as you can already see. It has very long setose legs, but also on the claws, it's very densely setose. But if you look more closely on an actual specimen of Trigobezoaris, a different genus, you see that it has some sort of fur coat. And it lives inside these very bland sea cucumbers, as you can see here down below, inside of its respiratory system, but also its digestive system. But I can tell you why it is that hairy. It probably has to do something with camouflage, but we still don't know, and we need more data on these animals in order to say that. An interesting rule of thumb with these crabs is that the dietary differences can play a major role in the morphologies of these animals. So, for example, the claw and walking leg morphology are oftentimes very much linked to their habitat, not their habitat, but dietary preferences. So, for example, this Holoteris has very huge claws, while these Amusioteris, a bivalve associate, has very slender claws, as you can see here. But why would you say that? So if you look at other body parts of these animals, so for example, the third maxillipedes, which are the last part of mouth parts, which they use to gather food with, of course, you see that A here, so this holoturian associated species, is very hairy and probably uses its mouth parts to filter feed their environment. While the other one, Amusioteris, probably uses its very slender claws in order to groom its host and just like grooms their claws again with their third pair of maxillipedes. So that way it can groom the host instead of filter feeding it. So they don't have these huge claws that they need to move to their face. An interesting extra character with this Amusioteris is that it is not bilateral symmetrical, but asymmetrical and with its walking legs, and probably has to do something with this weird fishing hook that it has at the end of one of the walking legs, that it uses its fishing hook in order to get mucus from outside of the shell, so outside of the bivalve, in order to get that also for his own food needs. An interesting extra character with these dietary differences is that a lot of these crabs have a cetal comb on their claws. So for example, here it's C2, you see this weird row of hairs on this Fabia species, also a bivalve associate. And if you zoom in with some photography, deep inside, well, not inside, but on top of these claws, you see that it has very weirdly shapen hairs and scales on top of its claws. So for example, here 2 and 3 are very short but very hard ridges on its claws in order to probably groom their host and groom their own legs as well. But we still don't know the true function of these different types of cetal rows, but it gives us an insight on how complex these animals are. So there are a few crabs. Well, I told you something about a few crabs where we know quite a lot about, but at the same time, we still don't know a lot about many more other species, some of which are really hard to explain their body shapes of. So for example, this Austroteres is covered with tubercles, as you can see here. So on below here is a lateral view of the carapace, and on top is the dorsal view of the animal. And for example, Durkheimia, another one you can see here on the side, has like this weird disc with a mohawk on top of its head, and we still don't know why it has it. Even weirder, it gets with Serenoteres. So Serenoteres lives inside these boring muscles and probably has something to do with their way that how they would get food and probably how they would get water down there, because they live inside these holes, of course, where the boring muscles has bored themselves into. So it has this weird roof-like structure on top of its carapace, and it looks almost like it has 2 faces and a pig nose, as you can see here. And the weirdest and my most prized CT scan right now is this Santasia. It is an inhabitant of the Tridacna giant clams, where it lives just between the folds of these animals. So it really doesn't seem to have a real use of these weird body shape. And you would say like, okay, the face and the back look kind of similar and it has legs just like the rest of them. But if you look on top, it has these swirly kinds of rows on the side of its carapace. So this margin is upturned and it has some weird tubercles on top of it. And we're just currently trying to figure out what is happening inside of this animal by using CT scanners to cut through it. So we now know that there's still a lot to discover about some of these groups, although they have been studied for more than 200 years. And we've decided to make a literature review, which was just published when I record this message, and was accepted in Diversity. So I'll probably send a link to all the people who are interested. So yeah, what now? Of course, we want to do more CT scanning of these animals in order to make a total picture of all of these different types of body shapes. We need to know more host associations in order to link that, of course, to their morphology. And we want to do more statistical methods, so using 3D morphometrics in order to quantify their shape data, in order to say something about the evolution from very basic to very complex in some of these weird species I just showed you. So yeah, I think that is it. So thank you so much. And are there any questions? I will be down there probably in the chat answering all your messages. See you.